Excuse me. So good afternoon, everyone. Welcome to our one of our webinars, part of our series in recognition of Gynecologic Cancer Awareness Month, which is September. Today we will have three speakers who are going to talk about their cancer research, gynecologic cancer research today. Um, just as a reminder, please stay on mute when you're not speaking. This webinar will actually not be streamed to Facebook Live because I haven't been able to get that to work, but it is being recorded and will be edited for YouTube later. And as our presenters go through their talks today, please use the chat box to ask any questions during the presentation. You can just ask them in the chat box, or if you want to ask a question anonymously, you can find my name, which is Aaliyah Poulos. It should say host next to it, so you know who to write a message to. If you'd like to ask a question anonymously, I will ask it on your behalf. This webinar series is part of programming from the Office of Community Engagement and Cancer Health Equity at the University of Chicago Medicine Comprehensive Cancer Center. As I mentioned, my name is Aaliyah Poulos, and I'm an education outreach specialist in this office. You can feel free to reach out to me and find my name on Zoom if you have any questions about our work or questions for our speakers. So our speakers today, we have Dr. Sarah Ackroyd, who's an assistant professor of obstetrics and gynecology. We have Dr. Catherine Kernod, who's also an assistant professor of obstetrics and gynecology, and Dr. Heather Whitney, who's an assistant professor of radiology, all at the University of Chicago Medicine. And they will be presenting their work today. I'm going to stop sharing my screen and throw it to Dr. Ackroyd, who's going to lead us off here. Dr. Ackroyd, thank you so much for joining us today and for being here.
Great. All right, you can see everything okay?
Yeah, it looks good.
Well, I just want to say thank you for having us today. I'm going to talk to everybody about early detection in gynecologic cancers. It's something near and dear to my heart and something that I hope will come to fruition during my career. So we'll start with just an overview. There we go. I have no disclosures related to this work, and I will be discussing investigational research. I do want to acknowledge some funding for some of these projects throughout the course. It would not be possible without all of these sources of funding. So the objective today is to review the prevention pyramid of disease. We'll also discuss primary and secondary prevention strategies in gynecologic cancers, and then we'll review new biomarkers in research and early detection. So as an overview, I'm sure you're all familiar with, but there are different types of cancers that we work on. And today we'll be focusing on uterine cancer, ovarian cancer, and cervical cancer. A lot of my work, or some of the work that we've done here at the University of Chicago, is more focused on ovarian and uterine cancer, but we'll highlight all three of them because it's important to know how to treat all of these and how we can get ahead of them. We know that uterine cancer, out of all of our gynecologic cancers, is the fourth most common for new cases. And you can see ovarian and cervical, as well as rare vulvar and vaginal cancers on the right. While it is more common to have uterine cancer, it is only the fifth, accounts for about fifth. That was the fifth most common cause of death in women who have cancer, followed by ovary. So we have a lot of work to do to try and improve these numbers. When we think about the disease prevention pyramid, we think about how can we intervene in a person's disease course to help primary prevent getting a cancer, detect it early so we can treat it completely, and tertiary refers to how can we reduce the impact of disease. A lot of the stuff that I'll be talking about today is going to be on primary and secondary prevention, but we will touch on some tertiary prevention because that is your typical care that you receive with your oncologist. And there's been a lot of developments in the oncology world as far as chemotherapies and new treatment strategies. So focusing on primary prevention, when we think about gynecologic cancers, we're thinking about cervical cancer is one of the best examples in the cancer world about how can you prevent disease. We have an HPV vaccine that prevents cervical cancer. We think around 80 to 90% of cervical cancers are HPV related. And then we have other risk factors, for instance, obesity, smoking, and healthy diet that can impact cancers such as lung cancer, breast cancer, and then uterine cancer for the gynecologic cancers. When we think about secondary prevention, we think about early detection. So, how can we find something in routine screening so that we can intercept disease before it becomes a problem? This could be anywhere from a biopsy to a surgical procedure that allows us to catch cancer when it is early. And so in the gynecologic cancer, that's cervical cancer, where we can do pap smears or HPV tests. As I mentioned before, tertiary prevention is more reducing the impact. In our world, that's going to be surgery, chemotherapy. How can we maintain quality of life and help take care of people so that the disease does not burden them? So what screening tests do we have in gynecologic cancers? So starting with the only one that we have pretty much in the gynecologic cancer world is cervix cancer. This is a picture in the lower right corner of what actually happens when you get a pap smear at your gynecologist office or at your primary care office. We're sampling the cells on the outside of the cervix to see if there's any abnormalities. When we think about preventing disease, that's going to be HPV vaccination, which is recommended as listed here for ages 9 to 45, and it can prevent cervix, vulvar, vaginal, and some upper ENT or head and neck cancers because they're also HPV related. The other secondary prevention is being able to catch it with a pap smear, where a gynecologist or a gynecologic oncologist or some specialist who's comfortable with these procedures is able to triage and figure out how can we stop this precancerous lesion from worsening. So it's important that people throughout their lifespan get this care so that we can help prevent cervix cancer. When we think about uterine cancer, and unfortunately, I'll say that for the rest of the gynecologic cancers, it's more about surveying for looking for different symptoms. We don't have a good screening test for a lot of these cancers, and I'll spend a lot of my time focusing on ovarian cancer. That's one of my interests in research, finding early detection, but uterine cancer is also a type of cancer that when we find it early, it's easier to treat, but we don't have a routine screening test to identify uterine cancer. So early detection is key, and increasing awareness of what symptoms are to look for. So any type of bleeding after menopause or abnormal bleeding are reasons why you should get medical attention. So when you usually see a gynecologist or your primary care doctor who's going to work up a concern for an endometrial cancer or uterine cancer, we're often going to do a biopsy where we sample the inside of the uterus. It's something we can do in the office. And we'll do an ultrasound, which you can see here on the right, just to visualize what your uterus looks like and if we need to be worried about any type of cancer or precancer. So although we don't have universal screening, screening for symptoms is part of the normal examination that you should expect from our primary care doctor or from your gynecologist. And if any of these things happen when you're in a time period where you haven't seen them, you're always welcome to come see a physician to get this worked up. So when we think about primary prevention, secondary prevention in uterine cancer, if we take a step back and prior to developing cancer, I'll talk a little bit about one of our uterine cancer research programs, and just want to highlight the people on the right, Dr. Diane Yamada, Dr. Jenny Fleming, and then Sandra Tillman, who's one of our data scientists. One of the projects that we've been working on here at the University of Chicago is using the data that we have. We're trying to understand what is it in someone's environment? What is it about their zip code, the environmental pollutants, or the stressors that they experience in their neighborhood? And how do some of these aspects lead to a higher rate of cancer development? And so this is just a picture of a map of the Chicagoland area. You can see this ADI refers to neighborhood deprivation. So what are different aspects in somebody's neighborhood on a zip code level that determine how many resources they have access to or how much pollution they have access to? So this is one of the projects that we've been leading over the past 1 to 2 years, funded through the Institute for Translational Medicine, who's allowed us to get kind of look at different environmental factors and see if patients who have had a cancer diagnosis if they had an equivalent matched person in the same environment or in a different environment, what in the environment could have contributed or been associated to them developing cancer? These are important questions because it's hypothesis driving, and we want to be able to get information so that other researchers in the environmental pollutants world and other aspects of primary prevention of getting a cancer diagnosis so that we can understand all of these aspects and help prevent cancer from happening. So that's an exciting project that's in process, and you're more than welcome. We have a nice video on this on the Institute for Translational Medicine website, and we're excited to share results as things continue to progress. So moving on to ovarian cancer, talking is probably one of the more challenging to diagnose in general, and in the sense that it's a rare cancer affecting about 1 in 67 and 1 in 70 women. And it's commonly diagnosed at advanced stages, so stage 3 and 4, due to our limited ability to identify early or vague signs of illness. When caught early, however, it can have a very favorable prognosis with 5-year survival greater than 90%. But unfortunately, there's a lot of space in our abdomen where our ovaries reside, and it's hard for us to identify these symptoms early. So no screening test at this time exists to identify ovarian cancer early. As an overview, this is an organization that has helped fund some of our work, Tina's Wish. It's an organization that provides grants to researchers who want to try and find early detection for ovarian cancer, and part of their mission is to increase awareness. I encourage you to check out their website. They have these postcards that you can send to anybody and print out and send, just to remind people what symptoms are that they should go and seek help. And this is a highlight of what are some of the symptoms that people can feel that should prompt them to get a visit with a physician. So when we look at screening, when there's a concern for ovarian cancer, you'll undergo a workup, whether that's at your primary care doctor's office, your gynecologist, or if you're seeing a gynecologic oncologist. Oftentimes we'll want to get some images to look in your abdomen and see, what do the ovaries look like? What do the nearby structures look like? Does it look like there's cancer in other areas? We also are able to use a blood test called CA-125. Many of you are probably familiar with it because it is a marker that we use throughout our disease course for ovarian cancer. It's a simple tube of blood, and they measure the levels of CA-125, which is a protein that the surface cells on the ovary excrete. So if cancer, by definition, is unregulated growth, if there are more of these cells, you expect that the CA-125 is going to be higher. I hope that this is a good test, but it unfortunately doesn't always perform well. And you'll probably see that when you talk to different physicians or your own oncologist, that sometimes it doesn't correlate well with either a diagnosis or in monitoring a cancer. So it's not very good at identifying early cancer and doesn't identify all cancers or even how to treat them completely. So it's a great tool to have, but it's not perfect, and we could do better. So generally the treatment is surgery and chemotherapy, and you can't biopsy. If a patient presents to you and they have a mass that is worrisome for cancer, with our limited tools of imaging, this blood test, CA-125, we are unable to, for instance, do a biopsy ahead of time to know, because we don't want to risk spreading a disease if we poke at the ovary or biopsy the ovary. So sometimes we have to do surgery to diagnose these and help get care as soon as possible. But surgery has risks, and we don't want to do 100 surgeries for every 2 diagnoses, but at this point in time, we don't have a better way to diagnose it, and so we have to do surgery oftentimes to get this diagnosis. So that brings me to, how can we find better ways to catch ovarian cancer? This is the main research project that I work on in the Langell lab, and it's the University of Chicago Ovarian Cancer Research Lab. This is a picture of the large lab that Dr. Langell and Dr. Kenny lead here at the University of Chicago. They've been doing this for over 20 years. The lab here is a comprehensive lab that works at trying to understand the biology of ovarian cancer. They want to understand why it formed, how do precancerous lesions become cancer, why does it spread, how does it spread, how can we prevent it from spreading? And they try to understand how do we find better ways to treat ovarian cancer and understand how chemotherapy changes cancer and how can we avoid cancer from becoming resistant to our treatment regimens. So this is a very large group, and one of the questions that Dr. Lingell and myself thought to answer was, how can we find a better way to catch ovarian cancer? And so when I refer to a biomarker, I'm referring to either a blood or a tissue test that we can use to identify a disease of interest. So for ovarian cancer, the question is going to be, number one, can we screen all women and see if there's something in the blood that can identify this early? And can we also find a way to decide who needs surgery and who doesn't need surgery so we can avoid the morbidity related to that and be 100% positive that we don't miss an early ovarian cancer? So our question is there a way to identify a blood-based signature to identify cancer in women? And just an overview of how our research question and our vision is to identify a blood-based biological signature that can identify cancer in women to use to both diagnose and screen women for ovarian cancer. This is a picture of an overview of what our research hopes to do. So anybody who presents and is going to have surgery for a mass, we approach and ask if they want to voluntarily participate in research in which we collect a tube of blood. That tube of blood is then used to examine if there is any signature, proteins, DNA, anything biological that is able to identify cancer versus no cancer. And we're able to correlate that with pathology because that patient is undergoing surgery already. We're really hopeful that this will be able to help us find a way to be able to identify cancer early. And I'm excited to see the results. I'm excited to see if we can help people and our population identify this disease early. So in conclusion, I think it's super important thinking about that initial prevention pyramid at the beginning is know the signs for gynecologic cancer and get all the age-appropriate screenings to try and catch cancer early. While we do not have good screening for ovarian cancer, innovative research and new minimally invasive methods, for instance, with a blood test, we are able to develop these to try and assess a biological status that may offer us an opportunity to identify cancer early. That's all I have for today, but feel free. I'm happy to take any questions at the end, and thank you for listening.
Thank you so much, Dr. Ackroyd. And we do have some questions coming in, as I mentioned. We'll save those for the end. Dr. Kernett will be speaking next.
Hi, everyone. I'll just pull up my slides. And Aaliyah, should I switch it? Does this look right?
That looks good. Yep.
All right, good afternoon, everyone. Thanks for having us. Thanks for being here. I'm excited to be a part of this kind of research-focused event for gynecologic malignancies. I think there's a lot of really important work, and some of it's being done here, and many of it's being done in many other places. So I'm Katie Kernett. I'm one of the GYN oncologists. My focus is in developmental therapeutics or helping find new drugs for patients, and I'm very involved in the clinical trials program at the University of Chicago, specifically for patients who have gynecologic cancers. So today I'm going to be talking about some of the clinical trial considerations, specifically for patients with cancer, and talking about some of the basics and background information I think is really helpful for people to know as they think about being involved in these systems. So I have no disclosures. I want to start off with a brief overview of clinical trials. This was from a nice website that I found that was talking about rare diseases, including rare cancers. And this just goes through kind of the big picture of what I want to discuss today. And this is the journey from how do we get from knowledge, just an idea, a concept, biology, how do we bring that all the way to the place where a patient can work with their oncologist and get that treatment for their cancer. And I'm going to be talking about it from a cancer perspective, but it's a similar process for any type of disease, any type of drug. So I'm going to start with this phase 0, and this is one that we don't often talk a lot about, but I think this is a really important context to have. And these, a lot of this information was gotten from cancer.org, if anyone's interested in learning more there. But phase 0 clinical trials or phase 0 research is exploring if and how a new drug may work. So this is kind of getting at the biology, the understanding of the mechanism. What does the drug specifically do in cells? What does it do with cancer? Why do we think it works? This can often involve patients, but it's usually at a very small number, and it's mostly an information gathering about the mechanism itself, not how well the drug works. So this is very few patients. We sometimes will call these like window of opportunity or other kind of translational work. The goal is to improve our understanding of the science of whatever the disease is. And one example would be taking a small number of patients and taking a medication, having them take a medication for a short period of time, maybe 2 weeks, and right before they're going to have a surgery, and then testing during the surgery what the tumor, what the actual tissue looks like. So it's not enough time for us to say, does this drug work or not? It's really enough time for us to say, what is the drug doing to the tumor to help us better understand how we can use it. These are not very common studies. There's very specific niches and criteria for when this is ethical and feasible to do, but this is an important part of sort of fact finding for some types of research. Phase 1 clinical trials, this is really where most people will start to hear about clinical trials. Like if you're talking with your doctor and they're talking about clinical trials, phase 1 is one of the more common types that people will talk about. Now, phase 1 trials are more time intensive, so not every hospital or location or doctor may be able to offer them, but they are very common across the country. And so the goal of phase 1 clinical trials is really to ask the question of, is the treatment safe? And so this is usually a small number of patients, but not so small that we can't get information kind of on a more standardized level. These are very rigorous studies. There's a lot of statistical planning that goes in. I would say an average number of patients, at least for the typical types, is somewhere between 15 and 45. It depends on the design. Now these phase 1 studies have what's called expansions, and so many of them are much bigger than that, but the true spirit of these phase 1s are that they're very small, somewhere on the order of 30 patients. The goal really here is to understand the side effects or the safety of the drug and to understand how the drug should be given in terms of dose. But I always tell patients, like the goal of any trial that we are choosing to do together, like a patient and a physician, is to find a treatment that works. So even though that may not be the primary aim of how the study is designed, none of us in the cancer world would do a study if we didn't have some hope that it would help a patient who's sitting in front of us. But the true primary outcome that we're looking at is about the safety. And so I tell patients, when you're watching those commercials and they have that whole long list of side effects that they say at the end, they're going through, a lot of that was gathered from these trials where patients receive a drug. And every time I see them, sometimes it's once a week or, once every few weeks, we sit down and we go through all of the symptoms or side effects that they have. And it's our job to try to sort through, based on what we know of the drug, what we think is related to the drug and what do we think is unrelated to the drug. And that sort of plays into that long laundry list of side effects that you see on package inserts, that you hear on those commercials. It's from many, many patients who have reported their symptoms to us, and we've cataloged them kind of standardly in these trials. So the example here, the classic design, is that there's a set of patients, usually start with 3. They're all given the lowest dose of the combination that we think would be safe and effective. And then we watch them for 1 month or 2 months, depending on the study, and we see how many of these patients have really serious side effects. If the answer is none, or whatever the cutoff is, then we say, okay, then after that 1 or 2 month waiting period, then we enroll the next 3 patients, and we say, okay, let's try the next highest dose. And we keep going until we meet pre-specified criteria. And then we say, okay, this is the dose of the drug that we should be using. And there's different, philosophies about how we do this, but that's sort of the backbone. Phase 2 is probably more along the lines of what people think of when they hear clinical trials. The phase 2 clinical trials, the question is, does the treatment work? And I would say, does the treatment work, parentheses, preliminarily? And this is usually a bigger study, 50, 100, maybe 150. This is a lower number of patients, but it's trying to see if the drug works, not just if it's safe, but if it's starting to work in a specific set of patients, whether it's a specific type of cancer or a specific mutation, some criteria that narrows this patient group down. We're looking to see if the drug might work there from a preliminary standpoint. So this would be saying, I want to look at patients with endometrial cancer, and I want to test this new drug, and I'm going to enroll, 35 patients based on the statistical plan that I made, and we're going to give all of the patients on this study this drug, and then at the end, we're going to see how many of them had their tumors shrink or go away, and how many of them didn't have any shrinkage or had growth of the tumor. And that gets at kind of this idea of, is it effective at treating the cancer? It's not usually looking at long-term outcomes. It's usually looking at kind of quick responses on imaging, but this is sort of the preliminary way that we decide, do we think that this drug is worth doing a bigger study in a specific subset of patients? And then phase 3, this is the one that I think is often the most interesting. It's the one that patients want the most because it's the ones that we know the most about. But there are some caveats about why phase 3s may not be, phase 3 trial options may not be the best choice for the individual sitting in front of us. A phase 3 clinical trial is really asking the question, is this drug, is this treatment better than what we're already doing? So not does it work, does it seem to shrink it, is it safe? But not only is it those things, but is it better than what we do? With the idea being that for how expensive drugs are, for how hard it is to, bring new drugs out, to do all the things to make sure it's safe, we want to make sure that it's actually better than what we're already doing. Because if it's not better in some way, either at treating the cancer or better side effects or better schedule, whatever it is, whatever we deem is most important, if it's not better than what we're already doing, then it is probably not worth the extra risk, the extra cost, all the extra burden that comes from having drugs that may not perform as well as we want. And so these are usually very big studies. So hundreds of patients, sometimes thousands of patients. They're very, very long. They're very expensive. They tend to be open more places because we are able to do, they're supposed to be more in line with what the experience would be if a patient were getting this in regular practice. So a phase 1 study requires a lot of blood draws, a lot of extra testing, a lot of checking to make sure it's safe. A phase 3 is a lot less of those things. It's more along the lines of what would we want patients to have to do if they were getting the drug regularly in the clinic. So for patients who have gotten carboplatin, for example, that study that looked at the phase 3 comparing carboplatin versus the older chemotherapy, they were hopefully using the doses of carboplatin that are still being used now, because these phase 3 studies help us decide how we use these drugs once the drug gets approved. So the study design here is that in order to prove that it's better than what we're already doing, we have to define what we think is the standard option to compare it to. And so this is where patients often ask me about, is there a placebo? There are very rare circumstances in cancer trials that a placebo can be used, and it's a very specific subset of these trials. In general, there's no placebo in cancer treatment because a study is only ethical if we believe truthfully in our hearts that both treatment options are good options. And so a placebo is not an acceptable option in most circumstances. So most of these do not have placebos. But what they do have is more standard chemotherapy. So there's a new drug and you might be comparing it to an old drug like paclitaxel or Taxol. Those it means that in order to know if this drug is better than this one, some of the patients have to get the old drug. So some of the patients have to be assigned to a chemotherapy or a treatment that already exists. And so for a patient who's really excited about doing a new study, the idea of potentially not getting the new drug can sometimes be disappointing. And so I think that's the downside to a phase 3. Yes, we have the most data to say that we think this drug will work, but some of the patients have to not get the new treatment. Otherwise, we will not be able to tell at the end of it whether it's better than what we're already doing. So kind of pluses and minuses there. But at the end of this treatment, this is at the end of the study, we hope to have information that can answer, is this better than what we're already doing? After this phase 3, then there's a lot of logistical things that happen from study groups and from drug companies and from a lot of other people who participate, but it ultimately goes to the FDA if the study was successful. And the FDA has their very rigorous process of whether the drug will be approved based on this data or whether it won't be approved. And most drugs, I will say, that start off at that phase 0 or we have a good idea, most drugs never make it to a phase 3, and very rarely would it become an FDA approved drug. So it's very, very difficult to get this far, but this is the goal. After the phase 3, if the data look good, we hope that the FDA would approve it. And it means that the patient who's coming to see me in the clinic doesn't need to go on a clinical trial to get access to that new drug. Phase 4 I put in here, this is it's more of a practical kind of consideration, but it's what else do we need to know about the drug? And this is generally patients being followed or being tracked after the drug is approved who are getting it in standard practice. So a patient sees me in the clinic, we just decide together that we're going to start this new treatment, but they might enroll on this kind of real world experience to understand what are the real side effects, what are the medications we can use to help with the side effects, how long do we give the drug, all these things that you can't necessarily answer in a clinical trial, but you might answer as you get more patients getting it in standard practice. And so this is where we are looking more at kind of symptoms and quality of life and other things that require much broader experience. Oops. And so I just put these in here. I like to share these with my patients when we're talking about clinical trials. The decision to go on a clinical trial is always very difficult. There's a lot of information to understand in a very short period of time, and often time is of the essence. And so these are some resources that I've found that are from reputable organizations, things that I've looked at that I think, hey, this actually has some really decent information that people can use. The last one down here is this clinicaltrials.gov. This should have every clinical trial that's being done in the US and often abroad. And it's one of the requirements from the FDA and from other regulatory bodies is that if you're doing a clinical trial that you list it here. And it's sort of our way to make sure that everyone knows about it, but also that we're being accountable and we're doing things the right way. So you can you can play around with this website, but you could type in like endometrial cancer and then look for clinical trials, and you can look in your area or other places around the country. And so what I'm going to end with is I talked about some of the logistics, but I want to ask the question, like, why should patients consider clinical trials? The reason I am very passionate about this, the reason I think it's really important is because it is a safer way to access new drugs and treatments. Drugs that patients are able to get in the clinic with insurance coverage and all of that were at one time a clinical trial. And so although I just said most of these drugs don't ever make it through all the way, it doesn't mean that there weren't patients along the way that benefited. So even though we talk in trials, we talk in statistics, we say, 50% of patients had their tumor shrink or use language that talks on broad scales. When I'm talking with a single patient in front of me, I tell them the only thing that matters is whether it works for you. Yes, we are gathering this information for the study, but even for studies that quote unquote fail or are negative, there are sometimes or often patients who still benefited from it. So we are still looking, these are ways to get newer treatments that are very, very tightly regulated. There's a lot of check-ins, it's a lot of safety checks to make sure we're doing it right. And then I put this up here as sort of a question, but there are some emerging data from gynecologic cancers and others that show that patients who go on clinical trials may have better outcomes. Is it cause and effect? I'm not saying it necessarily is, but there are some data that suggest that there's something about being on a clinical trial that may be helpful, regardless of the outcome of that clinical trial. This was a study that was actually published earlier this year. They looked at a big database of all the gynecologic cancer patients in there who got first-line treatment the first time they got kind of chemotherapy for their treatment. And they looked at whether they enrolled on a clinical trial or not. And they looked at ovarian, uterine, and cervical cancer. So this is just some of the data that they have. This is a very hard thing to interpret, but what I will say is that things on this side, things further to the left, these bars mean that the patients seem to benefit more from being on the clinical trial versus not being on the clinical trial. So patients who didn't ever go on a clinical trial, you can see their risk of progression is 1, or kind of the longer term survival is 1. But for patients who went on a clinical trial, they seem to do a little bit better. And that was true across different races. And this was true across different disease sites. And so although these data are hard for me to distill down very quickly, I think the take home here is that there does seem to be a benefit to patients in going on a clinical trial, trying these new drugs. Even again, if the drug doesn't work, there's something about maybe the more frequent check-ins or the kind of logistics of doing that may be helpful. So this should not be the reason to do it, but there are compelling data to say maybe this is something that at least patients should be offered and discussed. One thing that I thought was interesting, though, was in the study, they looked at about 860,000 patients, and only 880 of them enrolled on a trial. So that's less than 1%. I mean, that is a very small minority of patients who went on it. Now, the question is how many were offered and didn't go on it. But I think that in general, we can do much, much better. There are big trials out there. We are very motivated. We should find a way to bring trials to more patients. And so what I'll end with is kind of this question of who enrolls on these clinical trials. We know that trials change the way that cancer is treated. This is how we get drugs approved. This is how we get new treatments to patients. But they only represent the patients who are actually included on those trials. If we only do a trial in Japan, then we do not know if it works well for patients who don't live in Japan. And that is true kind of looking at all sorts of different characteristics. One of the big points of discussion is around race, and we know that there's very poor minority participation, and that is mostly related to factors on the physician side. We need to do a better job of making sure that there's access for every patient who wants to go on a trial. And it looked at this study, this phase 1 study did not have, none of the phase 1 studies had the enrollment that they expected for Black patients. And so this was a big position statement that came out that talked about ways to improve it. They basically said that our strict criteria sometimes make it harder. This is true for patients who live in rural locations, racial and ethnic minorities, older patients, other barriers that make it harder. And so how do we improve this enrollment? This is where I want to leave us. We do need to improve. There are lots of strategies in place to do this. There are many ideas about how to increase education, acknowledging some of the issues historically that we've had with trial enrollment in terms of like equity and justice and a lot of problems historically, involving patients, involving community members, involving advocates. And the big thing on our end is really making sure that it's offered, making sure that patients have the opportunity to go on it if they want to, and if they decline, then that's okay, but making sure that patients know it is out there. That's all I have. I look forward to the discussion after, and thank you for your time.
Thank you so much, Dr. Kernet. Dr. Whitney, looks
Thank
good.
you. And you can see it okay?
Yeah.
Great. Hi, everyone. I'm so honored to be here with you today, and especially I feel so lucky to present alongside my colleagues from OBGYN because a really exciting part of my work is collaboration with that department. So I'm pleased to see them here. And I'm happy to share with you a little bit about my work in AI of medical imaging for gynecological cancer diagnosis. I want to give a little background to my field because I come from the field of medical physics, and often it's kind of like in the background for a lot of the medical care that you might be receiving. So, medical physics, just so you're aware, is the study and application of the interaction of energy in the body for medical practice. And what this means in a nutshell is if we're putting energy into the body for some purpose, such as if you get an X-ray or a CT or an MRI, and we're putting that energy in to either see what's in there or treat what's in there, different things. That's where medical physics is a really strong player and an important contributor. And there's kind of four subfields of medical physics. You can kind of spot them here briefly. But what I want to point out to you is that from my role as a medical imaging scientist, we really play a wide role in terms of treatment using medical physics, diagnosis, which is what I'll focus on today, and then also a nuclear medical physicist. So your friendly medical physicists are behind the scenes whenever you're getting imaging done or having a radiation treatment done in other areas as well. So my research areas, you can just spot them briefly here, but I think a lot about medical imaging, how do we get more out of those images? And that's what I'm going to focus on in terms of my research today. Sometimes we need to actually propose changing an imaging technology for a purpose. That's a big deal, and we should have some really good reasons for proposing that. I work on how do we take an AI output. Let's say we do get to have an AI on an image and we present that to the physician for them to use. What is that like? What does that mean? How do they use it? When we are trying to put medical images, like tons, like thousands, millions of medical images together for different purposes, for big data medicine, how do we play a role in that? And then I also do some work in biomedical ethics, some questions that come up for AI medical imaging, as you might imagine. But I want to focus today on this top research question, how do we get more data out of existing imaging protocols? And I also just want to make sure everybody's aware of what AI means in medical imaging because it's a very, very hot topic term right now for a lot of different reasons, a lot of good reasons. And so I want to make sure you're aware, what do I mean when I say AI in medical imaging science? So generally with an AI tool, we're saying it can do something because it's been trained from data. So we can have computers do all kinds of things for us. In the past, it's been somebody tells the computer, do this exact computation on the image or with the data involved, like an equation is set up for that process. But with AI, what we're really doing is we still are doing some kind of task with the information, but we have trained the AI just on data itself. We have said to the AI, learn from this data, what is the best way to work with it for the end goal? And so we can use them on medical images for a wide variety of tasks, and that's a phrase called computer vision. So this is a whole field of AI. And we can do things like computer-aided diagnosis. So CADX is a term you may hear in some places. And it's actually been shown to be useful for diagnosis, prognosis. So prognosis is when we take an image and we can predict what will happen with the patient, how well will they do in the future. Or we can even on images predict how well a patient will respond to treatment. And this has a long history of effort, especially in breast cancer, and especially here at UChicago. And we're all now expanding these capabilities to gynecological imaging. And then you can do other things with AI. A couple of other things you can do are you can use the AI to detect a cancer. So is it there in the first place? Or to triage, to say, the AI has learned when to kind of order, when and how to order what the radiologist looks at, for example, how to get the most important, the most urgent things to the radiologist first. So we can do a lot of things with AI, and basically it's just expanding our clinical capabilities such that we're training these things from data, not necessarily a prefixed equation. And so one thing I like to emphasize in my work is that the ins and the outs are the same as they've always been ever since we had medical images when Röntgen discovered the X-ray over 100 years ago. A patient's undergoing imaging for some purpose, and then a clinician reviews that imaging as some part of their decision making. When we add AI in, again, the ins and the outs are the same. We have the patient undergoing the imaging, but what happens in the middle is that that image is evaluated through an AI. And I do want to emphasize here, we've heard a lot about the role of FDA in approving drugs, for example. Just like the FDA approves pharmaceuticals for their safety and efficacy, they also approve AI models that operate on medical imaging for the purposes of diagnosis. And I think that's important as a patient to know. There's a whole process by which you have to prove these are safe and effective. So let's say you've got one and it's in clinical use. Then the report's going to provide some kind of information about that outcome to the patient, and then they're going to use it for their clinical decision making. So the ins and outs are the same. The vision is that the physician is still the one making the decision in the end, but now they have additional information to work with. And something I want to emphasize is that using AI in medical imaging science is not new. It feels really new, especially because around in 2015 or so, there were some big advancements in computational resources and tools and kind of communities even. So it feels new because of that, but it's been around for a while, which is good, actually. This is a good thing. So one of the earliest papers dealing with kind of computer diagnosis on an image was actually published in 1963. And then one of the first, AI on mammograms happened in 1967. And then importantly, like I mentioned here at UChicago, it was one of the very earliest places to really move into neural networks. I don't know if you've heard that term, but that's a very common term right now about AI. And actually here at UChicago in the early 90s, they were one of the first places to use a neural network on a medical image. So AI in medical imaging is not new. And I think this is a really important point because it means that we're not trying to figure things out at this kind of really basic level. We have a lot to work with, and that actually inspires my work. We have a lot to work with, and we can put things together in the best ways possible. So I'm going to focus on my research in AI of medical imaging with ovarian cancer. And I especially want to just highlight what Dr. Ackroyd said. We do not have good image-based screening for ovarian cancer right now. And people have been trying very hard to do this. There was a major study in the UK, for example, which studied the potential for ultrasound imaging as is to serve as an ovarian cancer screening tool. And the result of that study was unfortunate. It said there's no evidence that it improves mortality in patients. So that was a disappointing study, but we learned from it. And we said, we got to rethink some things. So that's really inspiring my work. How can we think about medical imaging eventually as a screening tool? But right now we're focusing on some diagnostic tools on ovarian cancer from the imaging, and then we'll apply that to what we learn. We'll apply what we learn to screening in the future. But for now, so we've had a lot of great introduction to ovarian cancer. And so here, this slide is recounting a lot of things that you've already heard, but they really inspire me in my work about the potential for how do we get more out of our images and help with this. So you've heard about how the incidence is quite low, but when there is an ovarian cancer, it has a high impact, a high likelihood of mortality. And not only that, the adnexal lesions, these ovarian lesions are very common. So over 200,000 women undergo surgery for a pelvic mass annually. And transvaginal ultrasound is a really key player at diagnosis. So it's relatively inexpensive and quick. However, there's this trade-off. It's easy to use, but the interpretation is largely qualitative, and there's lots of variability in the acquisition. So these are some things we're looking at as we think about applying medical image science to what we can do better. So, our vision for AI preoperative diagnosis of adnexal masses on ultrasound images is really laid out in this way, especially because of our collaboration with our OBGYN faculty here. They tell us the woman undergoes the imaging, and the clinicians tell us they want to be interacting with the image as they're finding out what the AI thinks about it. So the clinician draws a box around the mass that they're interested in, and our AI automatically outlines it, as I'll show you, and reports a likelihood of malignancy, and then the physician can use it as part of their decision making. So here, I wanted to share with you some basics of our recent study that or studies that we've published on this, just to kind of let you know what's behind the scenes in an AI tool and what are our results. So, I mentioned computer vision, AI of medical imaging. It's quite mature. We have a lot of things we can work with. So we actually combined several different AI tools kind of in a chain to create a pipeline that it takes the image in, then we have the box from the clinician, and then it reports that likelihood of malignancy. And it does a few things automatically. You can kind of watch it here on the right. We start with a full image. We have an ROI, a region of interest from the clinician. The UNet, which is one of our AI tools, does an automatic outline here in pink. The computer creates 2 additional views of those images. So this is like a filter. If you think about putting a filter, maybe, in glasses that have like, sunglass dimming, you can maybe make some lenses that do some additional things with what you're seeing. That's the same thing here. We have 2 other ways, the entropy filter and the standard deviation filter. And then we do another AI. And basically, in the end, what we get is the ability to kind of carve out on the image automatically these different components of the mass. Because our hypothesis was that, maybe different parts of the mass are important for making that automatic prediction more than others. So the result is that we found this to be very effective. So this hybrid AI tool, this is a figure that shows the results. And the takeaway for you is that we made this measurement called an AUC. And if the AUC is 1, it means the AI did a completely perfect job. It can go from 0 to 1. So our AUC on this tool, when we fed in images of masses that in the end result, we knew what the end result was, but we could kind of train the AI to take the data in and learn from it. We got an AUC of 0.93, which is really quite good. This was really quite effective in this data set. It's a small data set, so we're expanding that a couple of different ways. We're also trying to get more images that we can put in, but we were really happy about this result. And so here's just some examples too about what we can kind of see through the AI. These are a number of different masses. And again, our AI kind of automatically outlines. It does these 2 additional views, combines that together to get these 2 different kind of subcomponents, if you will. And this was really informative to us to kind of see these lesions just a little bit differently and learn from them in additional ways. So we have 2 recent papers, and I have screenshots of them here because if you look at the author list, a big important takeaway of this is the collaboration between departments. Dr. Ackroyd talked about how they have a study where they do biomarkers and they're consenting patients. One of those studies also consents the patients for us to be able to use their ultrasound images, which we're really grateful for. So you'll see a list of people here who are for both radiology and from OBGYN, and that's the big takeaway here, is that collaboration, how important it is. So I'll wrap up by thanking my lab. These are my lab members here. They're amazing. I also collaborate with the Langell Lab, a great team there. And we're really excited about our funding sources that have come together to help support this work even further. We're really, really grateful for that. Thanks so much for the opportunity to share with you our work and especially to highlight the collaboration between departments. It's really exciting. Thank you.
Thank you so much, Dr. Whitney. Okay, so we did have quite a few questions come into the chat. In the interest of time, I'm going to choose a few of them. Some of them also came in to me, as I mentioned, privately. So one question here is, how can AI imaging um, improve patient outcomes, but also reduce the costs of healthcare? So I guess that would be more for Dr. Whitney. How can AI imaging help to reduce the costs of healthcare?
Well, I think the one thing we want to make sure doesn't happen is we don't want to reduce kind of the benefit of or the performance of using an image. So in terms of reducing costs, there are some interesting models that are happening where if you've got a situation where traditionally two radiologists have read an image, if you're thinking about cost in terms of those two radiologists, one model is to have the AI be the second reader. Now, I wouldn't say it reduces cost long term because really what that opens up is the second radiologist can go do more interesting things. But that's one way of looking at it, is it may kind of shift who's doing what when. And part of that benefit, again, maybe it not be a dollar exactly, but kind of makes the work better for radiologists and makes them more high performance in their work. So we typically in my work don't tie the use of an AI to dropping costs, but to increasing value overall and getting a better result for the patient.
Okay, there's another question here for you, Dr. Whitney. How can we make sure that women in low resource areas get the same benefits from AI screening?
That's a really great question. And I want to highlight too, that's what a big inspiration is behind this ultrasound project in the first place. Because you can go to more advanced imaging, such as MRI or CT. And let's, say if only MRI was available for a certain type of ovarian imaging, it would definitely be outside of the reach of many women, just for a number of reasons. But if we can build great tools around ultrasound in particular, since it's so easy to use, so inexpensive in many ways, it really keeps that kind of imaging input more equitable. And so actually, Dr. Langell and my collaborators, he always says, let's make sure this tool can be used anywhere. And it's a big inspiration. So that's why we focus on ultrasound, and that's why we focus on AI, because it can kind of level the playing field about who's looking at the image and when and how well they do.
Um, so there's a question here that came to me for Dr. Kernet, asking about the relationship between pharmaceutical companies and researchers when developing clinical or when doing clinical research and clinical trials.
Yeah, that's a wonderful question. And I think it gets at a lot of the considerations for the drug development process. So when we think about a clinical trial, one of the first questions to ask or that we all have to consider is who is funding the trial? Where is the trial coming from? Who's designing it? Who's paying for it? There are different versions. Many of them come through the pharmaceutical companies themselves. They design it, they have a drug, they set it up, and then they reach out to people like us and say, hey, we'd like to do this clinical trial. Would you guys be wanting to participate with your patients? There's other kind of mediators or kind of like in-betweens, these what we call cooperative groups of usually oncologists, in our case, gynecologic oncologists, who get together and help decide kind of what are the drugs, what are the patient populations, what are the things that are most important, and help kind of broker it with drug companies directly. And so it's sort of like a hybrid version. And then there's ones that are more like these investigator initiated trials, like out of a lab, like one of the labs I work with, they come up with a good idea and then we propose it to a drug company, or we find a way that we get different funding, but it comes from ideas that we had. So there's different ways that the trials can be funded, but it usually is either from a pharmaceutical company or from like NIH or kind of research funding primarily. But it's a talk for a whole other time, and I think there's a lot that plays into what that means for the trial design and development and things like that.
Thank you. This next question, I think it could be Dr. Ackroyd or Dr. Kernan. I'm not sure. It says, what role do you see immunotherapy or targeted therapies playing in the future of gynecologic cancer treatment?
I can answer from like a trials perspective. All of these new treatments, immunotherapy is a very big one. We have a lot of clinical trials that have come through looking at old immunotherapy agents, new immunotherapy agents, different combinations, and the same is true for targeted therapy. So I think that many of us in the drug development world think that this is going to be the future of kind of personalized medicine using these non-chemotherapy approaches. Of course, chemotherapy works some of the time very, very well, but in the times it doesn't work, finding these alternative options like immunotherapy and targeted therapies are going to be really important. So I think that there's a lot of hope and promise and even approvals that have already happened in the gynecologic cancer space.
And then Dr. Ackroyd says, how can research in biomarkers and genetics be better translated into everyday screening for women?
I think that's a great question. One thing I did not highlight is the
One second. Someone's in. Sorry, everyone. One sec.
There we go. I was going to say one thing I did not highlight, it's a great question, is that there is genetic testing available for there specific guidelines that the NCCN recommends where if there's any sort of family history of cancer, colon cancer, breast cancer, ovary cancer, pancreas cancer, whole list. And if you've been affected by one of these, getting genetic testing can help increase your awareness. While I had mentioned that there is not good screening for ovarian cancer, those who have the BRCA gene, the breast and ovarian cancer gene, are at a higher risk for breast and ovarian cancer. And we follow them annually with specific ultrasound as well as CA-125. And although historical data has not shown that it can catch cancer early, it is a means for us to have close contact with people to help see if we can identify things early.
And then I had one more question here that came in to me again for Dr. Ackroyd. Do you foresee a screening test for other types of gynecologic cancers besides cervical in the near future? I don't know if that's an answerable one, or I don't know what you think.
Yeah, I think that's the hope, and that's sort of why myself and Dr. Lingell lead this study. We're hoping that we can use, there has to be a signature somewhere in our biology. It's just what sort of technology has come along that's allowing us to identify these things. Going along with what Dr. Whitney said, we now have AI tools and supervised learning, and we have so much improved strategies to identify molecules and proteins and DNA that we were not able to identify 10 years ago. So we're pretty enthusiastic about being committed to trying to find a signature so that you start with one cancer, we hope, and there are many companies also working on finding multi-cancer gene testing panels. So our benefit here is that we have expertise in ovarian cancer and we have the patient population that we're able to help use that to try and develop these technologies. And I hope that that will definitely be within my career and hopefully in the near future.
Awesome. Thank you so much. I think that's all the time we have today for our questions. As Dr. Ackroyd mentioned, genetic testing, I did drop a link in the chat box for the Cancer Risk and Prevention Clinic, which does offer testing really easily through the University of Chicago. So just check out that link in the chat box. Okay, huge thank you again to Doctors Ackroyd, Kernet, and Whitney for sharing your time and your expertise with us today. We really appreciate it.